Guys, here's the stuff for your biology test on mitosis and meiosis. The difference between the different states of DNA is very subtle. You'll see these vocab words over here all relate to what's happening with DNA. Chromatin is the thin, noodle-looking DNA in its relaxed, uncoiled form. Kind of looks like this, and that's what we see at interphase. Chromatids are dense, coiled, compacted DNA. They form half of a chromosome. We'll see those in anaphase of meiosis. Chromosomes are two identical chromatids. They're joined at the center by this little button here called a centromere. Sister chromatids are two identical copies of the one you got from mom. So this is the one that's in all of your body cells. And then in interphase, it doubles. So now you've got a complete sister chromatid pair forming a chromosome. A tetrad is when you get 4 total chromatids. You've got the one from mom, the one from dad, each of those doubles during interphase, so you have 1, 2, 3, 4 chromatids, making a tetrad, a double from mom and a double from dad. Diploid is a word that describes a cell that has 2 copies of every chromosome. So when you were doing karyotypes and you were sorting out all those chromosomes, you found that there were 2 copies of everything. That cell was diploid. That's our body cells. 23 pairs for a total of 46 in humans. Haploid is what we use to describe our gametes, or our sex cells. Sperm and eggs have 23 chromosomes, no pairs. 1 copy of each chromosome means it's haploid. This last word over here, centriole, this is the target or the anchor for our spindle fibers. It's going to move to opposite ends of the cell, and it's going to pull all of our chromosomes toward those opposite ends of the cell. 4 stages of the cell cycle. We start with mitosis. The rest of these are part of interphase. Remember, that's where cells spend most of their life. G1 is where they do growth in their normal life, and they prepare for the S phase. The S phase is where they double their DNA. After they've done that, they go to G2, which is the last big growth phase before we divide them in half through mitosis. There are checkpoints at each of those. So the M phase, we have the metaphase checkpoint, that buddy check, making sure that we have the correct number of chromosomes. G1, we check to see if the cell is healthy enough to actually continue on. The S phase, we check to make sure that there's no errors with the DNA. And then the G2 phase, we make sure that they haven't grown out of control or anything like that. If they fail one of those checkpoints, they should do this thing called apoptosis, which is death. They're programmed to die if they don't pass a checkpoint. But cancer cells avoid that, and they do continue going, and they grow out of control. Here are the stages of mitosis. I puked on my algebra teacher's car, so interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. In general, we start with chromatin, loose DNA, uncoiled DNA. Cells spend most of their life here. Then they go into prophase. Their chromosomes start to condense. Metaphase, they meet up at the middle. Anaphase, they're going to be pulled to opposite sides. Telophase, we wrap them up with new nuclear membranes. And then cytokinesis, they're completely relaxed. We split them in half to form 2 daughter cells. In animal cells, the cell does pinch inward, just like this, and that forms what's called a cleavage furrow. In plant cells, they can't pinch inward because their cell wall is too rigid, so instead they build a new cell plate right here across the inside, and that's how they divide. In meiosis, we've got the same phases: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. And then we skip interphase and start meiosis 2, prophase, metaphase, anaphase, telophase, cytokinesis. What's unique about this is that during prophase 1 and metaphase 1, we have crossing over. So here's your tetrad, 4 chromatids, double from dad, double from mom. During prophase and metaphase, they swap little pieces of DNA. So now this chromatid is a mixture of what you got from dad and what you got from mom. This is a mix of what you got from mom and what you got from dad. That's going to randomize what possibilities we could create down here when we actually make sperm and egg cells. It's a good thing because it increases our genetic diversity. So we start with 46 in all of our body cells. During interphase, the S phase, we're going to double that number. We're going to end up with 92 chromosomes. 92 chromosomes continues until anaphase. They start to split and move to opposite sides of the cell equally. So we're getting 46 in each of these cells at the end of cytokinesis 1. We skip interphase so that chromosome number does not double, and that's how we're going to divide it in half and continue on until we get just 23 chromosomes in each of our sperm and egg cells. That's important because remember, if you had too many chromosomes, you usually have problems with the offspring. So why is this whole meiosis 2 necessary? Why can't we just do meiosis 1? The reason why is because we have to cut the chromosome number in half. and then crossing over right here, this occurs and gives all of our sperm and egg cells a slightly different mixture of DNA. So you could have 4 egg cells and have quadruplets, 4 kids at the same time, and have them all come out looking different because they're all inheriting a different mixture of these chromosomes. So the difference between mitosis and meiosis: mitosis occurs in our body cells. So a good way to remember it is mito, spell the word toe, cis. So it's all about your body cells. You're going from one diploid cell to another diploid cell, 2 copies of every chromosome, 2 copies of every chromosome. They are identical clones of each other. At the end, the daughter cells look exactly the same. They have the same chromosome number, the same information. There hasn't been any crossing over, so we haven't scrambled the DNA. In meiosis, we're going from diploid to haploid cells. The chromosome number is cut in half, so we went from 46 to 23. We're mixing up those chromosomes so these cells are no longer identical, and once again, we're making sex cells in this process, either sperm or eggs. So advantages to reproducing by just doing mitosis: it's not so complicated. You have a lot fewer stages, and you don't have to find a partner to make a baby. You just split in half, and you've created more of yourself. Negative is that all of your offspring are clones, and they'll have the same DNA. So if you're allergic to peanuts, all of your offspring would be allergic to peanuts, and if they encounter peanut butter, they would all die, and that would not be good. So what are the advantages and disadvantages to sexual reproduction through meiosis, which is how we do it? All animals, all plants do it this way. We increase our genetic diversity. So if you're allergic to peanuts, but your partner is not, there are chances that your kids are not allergic to peanuts. They could survive the giant peanut butter outbreak massacre. Negative is that it's really complex. There's lots of steps, and you do have to go find a partner or a mate in order to produce a baby if you are reproducing sexually. Haploid and diploid. I know you guys are a little confused about this. So in a normal body cell, we start off with 46 chromosomes in all of our body cells. So a skin cell, a muscle cell, a nerve cell. 46 chromosomes are there. Then, after DNA synthesis during interphase, so in this step right here, the chromosome number has doubled. We have 92. And that number continues all through here, and then it starts to split in half. So at metaphase 2, down here, arrow went crazy, sorry. Number of chromosomes at metaphase 2 then is going to be 46, because we've cut that number in half. Went 46, 92, 92, 92, 92, now 46 here, 46 here, 46 here, 46 here. So that's how that number split in half. And then at the very end, way down at the bottom, we cut that number in half again. So we had 46, now it's 23, 23, 23, 23. So that's how we get to our haploid number. And this pattern exists for all living things. So you start with a number, you double it during interphase. You cut that number in half in meiosis 1, you cut it in half again during meiosis 2. Start with a number, double it in interphase. Cut it in half in meiosis 1, cut it in half again in meiosis 2. A karyotype is what we did with our online lab. It's a map to organize all the chromosomes into pairs. We sort them out by size and then we number them. So for example, these two are the same size, they have identical information. They're a pair of chromosome 1. Here's a pair of chromosome 2. If you have a male, they will have a Y chromosome. They could also have 2 X's and a Y, still a male. The Y chromosome is the one that codes for testosterone and gives you male characteristics. A female is going to have X chromosomes. They're female if they have 1 X, 2 X's, or even 3. As long as there's no Y chromosome, it is a female. So in this first chromosome, this is normal. This karyotype shows no extra copies, no missing copies. If it's XX, that would be a normal female. If it's XY, it's a normal male. This one, we look through, and yeah, some of them look more crooked, but we still have a matched set. There are 2 of every type of chromosome until we get to chromosome 13. There's an extra, so that indicates an abnormal karyotype. And the fact that there are 2 X chromosomes means that this is female. If it had been 1 X chromosome and 1 Y, then it would be a male. Stem cells are blank cells that have not differentiated. They haven't been programmed to become a specific type, so they can be used and programmed to become any kind of cell or tissue. So because of that, we can cure or heal diseases like Alzheimer's. We can make those stem cells go in and become the damaged tissue. We can repair tissues, organs, heart attack scar tissue. We can heal injuries to the spinal cord. We can have organs be transplanted into people that match their actual body. And all of those are good things. The bad things are it might be an ethical or a moral issue for a lot of people as to where we're getting these stem cells. Some of them are embryonic, some of them are from adult tissue. It's promising research, but we haven't proven it effective yet in a lot of human research. And it's very expensive, not only because of where these tissues come from, but because of how limited we are in our ability to use them. And then the last one is that we do have some unpredictable side effects. It's been warned that you could actually end up getting cancer from stem cell treatments or other abnormal cell growth. That's it for the test. If you have other questions, the answer key has been posted and emailed to you, so you can check the digital version there, or you can look for your notes. Email me if you have any other questions.